talk of the day. Thank you, judges, for showing up, and miscellaneous people. So I'd just like to briefly introduce Alex and Gus. They've been doing research. Gus has been doing it with me for almost a year now, and Alex for almost 2 years. And so they are going to talk to you a little bit about some of the spider submersion behavior stuff that we've been doing in our lab. So
Thank you.
take it away.
All right, so to begin, I'd like to get a quick show of hands for how many people can say that they prefer the company of spiders. Of course, not many. So then you've probably all heard the expression or someone yell, "Flush it down the toilet," after they see a spider in the house, right? Well, we're here to tell you that spiders can actually breathe underwater. Hello, my name is Alexander Mullins. This is Augustus Forrest, and today we'd like to talk to you about morphological spine characteristics contributing to submersion tolerance in 2 species of real spider. So to give you a little overview, we'll first be talking about some of the selective forces that have driven this behavior. We'll then go into some of our study system and what we did, and as far as data collection analysis, and then some of the data, and then some concluding remarks and discussion. So organisms interact with their environment, and this is what ecology teaches us. And the environment subsequently exerts selective pressures on these organisms. These selective forces come in 2 forms, biotic and abiotic. Biotic are living forces and include things like local competition, where organisms in a local area compete for resources, as well as predator-prey relationships, where predators seek to become more efficient in prey capture as well as intake. And prey species aim to reduce the effects of predation. Abiotic factors are non-living factors and consist of things like humidity, temperature, and just overall weather patterns. So, Okay, so now that you got an idea for selective forces, we'd like to take you and introduce you to our study system. So, in our study system, we have a small riparian corridor where both of these species coexist. We have Pardosa valens, which is a small, strongly aquatic species that can be found foraging in the aquatic interface, where it actually forages on insects trapped in the surface tension of the water. Rabidosa centrida, on the other hand, is a much larger species and is less tied to this aquatic interface, and can be found kind of on the upper limits of this riparian habitat. So both these species are found within the same local area, but their selective pressures are much different. Biotically, we have the main predators of Rabidosa centrida being primarily birds and lizards. Alternatively, for Pardosa valens, we have it falling victim to mostly predation by Rabidosa centrida, and actually consists of about 80% of their total diet. So to avoid or reduce the effects of predation, Pardosa valens has evolved the ability to dive under the water, and Rabidosa centrida still needs to eat, right? So they too have evolved this ability. However, not quite as specialized as Pardosa valens. So here is our study system on a typical May to June day, where the stream is relatively low. And this is that same local area during the monsoon season in July and August. So we can safely assume that these species are affected by this increase in water. And this is an example of our abiotic selective force. And the fact that Pardosa valens inhabits this inner creek channel means that they're probably more affected by this increase in water or flooding event than Rabidosa, who would inhabit this upper riparian area. So I've talked to you a little bit about submersion tolerance, and for those of you who are not quite familiar with this behavior, I'd like to spend a moment to further discuss it. So submersion tolerance is just the ability to resist drowning, basically, or tolerate submersion. In this case, aquatic submersion. The mechanism by which these spiders, as well as many wetland plant species and insects do this, is by having a cuticular surface covered in hydrophobic hairs. This facilitates the creation of an air bubble, which subsequently can be used for aquatic respiration. This is an example of our diving bell spider, which is kind of the poster child for aquatic respiration exemplified in spiders.
So after observing the submersion tolerance behavior, we ended up developing two questions. So first, is there a difference in submersion tolerance between gender and then between species? And then second, what morphological characteristics actually affect the submersion tolerance? So moving on to our methods, we started with a collection in Cave Creek, which is in the Chiricahua Mountains of Arizona. For P. valens, we collected 38 total, 17 males and 21 females. And for R. centrita, we collected 42 total with 17 males and 25 females. The first test that we ran was the aquatic running speed, which you can see here and here, with P. valens up here and R. centrita down here. So how we did this is we took a PVC pipe and we cut it horizontally. We then capped the ends and then marked out centimeter increments, as you can see here and here. We then filled it with water and placed the spiders on top of the water and then chased them down the raceway, and then measured how far they went in centimeters per second and then converted it to meters per second. So as you can see here, P. valens is quite a bit faster than R. centrita. It goes through 3 full rotations before that one even completes 1. So the next test that we ran was the submersion tolerance test. So what we did here is we took these centrifuge tubes and we placed the spiders in the centrifuge tubes. We then put a cotton ball, which you can see here at the top, and then filled it slowly. We filled the centrifuge tube slowly. This allowed the spider to create the actual bubble and allowed there to be no air at the top. We then left these spiders for about 1 hour. We then dumped them out and set them on their back, and we measured their time to right. So what this means is we waited until they flipped all the way over and could move away from us. This basically shows, it's basically resuscitating the spider, so that at that point it could move up. The reason that we chose to do time to completely flip over rather than just move is because from an ecological standpoint, you would believe that the spider wouldn't just have to move to get away from its prey, it would have to actually move away from its prey. So next we moved into our microscopy investigations. We did these using image J, which allowed us to measure these images that we took. On each of the spiders, we took the 4th leg, which is the farthest leg back, the closest to the abdomen. The reason we took the 4th leg is because we believe that it's used in the creation of the actual bubble that allows the spider to breathe underwater. So for this to work, the leg and the abdomen would have to work together to actually form the bubble to encase the book lungs, which are here and here. So those are the respiratory surface of the spider, so that has to be completely encased to allow oxygen to diffuse. So on the microscopy investigations, we first looked at the average femur spine density, which is shown here in blue. Relative femur spine length showed here in red, as well as femur spine angle showed here in green, and then finally the abdomen spine density, which you can see here.
So, moving on to results. So what these results show, first showing our survival plot, which shows that the proportion of non-righted specimens on the y-axis and time to right on the x-axis. Proportion of non-righted specimens, with 1 it being that no spider righted themselves, so it's a little bit opposite. No spiders righted themselves, and then 0 being that every spider righted itself. So we then have different letters signifying significantly different groups. So A and B are the 2 groups here. So when looking at A, or the green line, we see that it corresponds to female Pardosa valens, which had a significantly more rapid time to right than all of the groups tested, including males of the same species. So although the males of the same species did not right themselves as fast, one thing I'd like you to know is that the same proportions of Pardosa valens righted themselves by the end of the submersion trial. So roughly 90% of Pardosa valens species righted themselves by the end of the trial. So moving on to Rabidosa, we have B, which includes these 3 groups, and Rabidosa being the black line and the red line. So here we see that there's no sexual dimorphism or difference between submersion tolerance between these 2 groups, or male and female, and that roughly 80% righted themselves by the end of the submersion trial. So now I'm going to take you through a few graphs, figures that very closely resemble each other, with our dependent variable here on the y-axis and our groups tested on the x. First, we have aquatic running speed measured in meters per second. So here we see our Pardosa valens. Once again, our letters indicate significantly different groups. So we see that Pardosa valens as a species is significantly different from Rabidosa centrita as a species. And there's no intraspecific differences between male and female here. Now looking at relative femur spine length, we see once again that Pardosa valens is significantly different from Rabidosa centrita as far as femur spine length. Next, we have femur spine density. Here, once again, the trend follows with Pardosa valens being significantly different from Rabidosa centrita. However, this time we also see sexual dimorphism in this trait, with males having a significantly higher spine density, femur spine density, than females of the same species. And then next we have femur spine angle, and here we did not see any significant difference between any of the groups tested. And lastly, we have average abdomen spine density. Here, once again, we have Pardosa valens being the victor, and Rabidosa centrita having a lower abdomen spine density, and sexual dimorphism exhibited between males and females of the Pardosa species.
So now, kind of what does all of that mean? So moving on to our survival analysis, we see species differences morphologically. So as you can see here, here's the Rabidosa species compared to the Pardosa species. And as you can tell, the Rabidosa species is quite a bit larger than the Pardosa species. So what we see here is that a paper done in 2003 by Tafova and Tuff showed us that size was actually detrimental to this submersion tolerance. Just because their surface area to volume ratio, they have a lower surface area to volume ratio, meaning that they are going to have a harder time with the submersion tolerance. Another study that we looked at in 2009 was done on the specialization of 3 different species of wolf spider. So what we think is that this compares to ours is because some of their wolf spiders actually withstood flooding events, so they would actually take it and hold on to something or just move with the water downstream, compared to another species which actually evaded it or tried to get up on vegetation or trees and stuff like that. So what we think is how this compares to ours is that the Pardosa species would actually be the withstanding species, just because of how close it is related to the water and where it is in the stream bed, compared to the Rabidosa, which we believe would be evading, trying to get up just because it's a little further from the stream bed. Next, just strictly looking at the female P. valens, they are physiologically dimorphic when it comes to metabolic rate. So why metabolic rate is important is because the higher your metabolic rate, the faster you burn through oxygen. So when they have this bubble, you can see that they burn through the oxygen faster with a higher metabolic rate and then with a lower metabolic rate. So these females actually have a lower metabolic rate, which we could see in the that submersion tolerance graph, with them being the first to write or the fastest to write. So a study was done in 2006 by Walker and Irwin, where they looked at 2 comparable species to the species that we looked at, so Pardosa novina and then Hognahelio. The Pardosa obviously being similar to our Pardosa species, and the Hognahelio being similar to the Rabidosa. They also found that the female Pardosa had a lower metabolic rate than the male, and that Hognahelia, similar to our Rabidosa species, didn't have any differences between them. We believe this is because of certain selective pressures that are on the 2 larger species of spider. Then moving on to our morphological spine characteristics. So relative femur spine length, we found that P. valens had a greater length than R. centrita. This is most likely a morphological adaptation, possibly due to how much contact P. valens comes into the water compared to R. centrita. Since P. valens is a little deeper in the stream bed and closer to the water, they're going to come in more contact, so they're going to need longer spines. The average femur spine density and average abdomen spine density, we found that male P. valens actually had higher density than female P. valens. This could possibly be sexual selection, so it could almost be like a handicap hypothesis. So much like certain birds and stuff like that have really long tails and stuff like that, and although it lowers their fitness, they still have to do it to attract a mate. We believe that we're seeing this in the male spiders. The females are selecting for the males that have a higher density in spines. And then the average femur spine angle, although we didn't find that it was super significant, we think this could be because we took the spine angle post-mortem. So spiders use hydrostatic pressure to actually throw their legs out. That's how they use their legs and stuff like that. And these hairs are actually seated inside of the body. So what we believe is, since we took it post-mortem, there was no hydrostatic pressure, meaning that the spine angle was somewhat irrelevant. So in conclusion, we see major differences in the species with size, metabolic rate, predation pressures, as well as the relation to the aquatic interface. But most of all, when it comes to submersion tolerance, we believe that size is playing the biggest role. So since the Santrita is so large, it's not doing as well with the submersion tolerance. Then when it comes to sexes with the Pardosa, certain selective pressures push towards the metabolic rate. So what this basically means is that the females are just, for the Pardosa, are just going out to hunt. Well, the males are having to go out and hunt as well as try and find a mate. So with that increase in expenditure of energy, the male is going to actually have a higher metabolic rate, meaning that it's going to burn more oxygen when actually forming the bubble, compared to the female who's just going out and hunting.
So next time you think about flushing a spider down the toilet, you might want to think twice. Hope he doesn't have your address.
We thank Matthew Stephenson, Chris Brown, John Whittinger, Bob Baggett,
as well as Adams State University, Tennessee Tech University, the American Museum of Natural History, and the Southwestern Conservation Corps, or a research station for their contributions and facilities.
Here's our literature cited. Are there any questions?
So, do you think of any almost engineering applications for this? I mean, obviously, what we see in the natural world, if we can replicate it more artificially, might have some useful ends. Is there any application beyond the domain of biology for this?
Sure. So the mechanism by which these guys create the surface bubble is hair, right? So the hair doesn't allow for oxygen to be trapped in that kind of the interstitial space between the leg and the tip of the hair. So if you think about maybe, I don't know, suits, if we do some sort of like a swimsuit, has the diving suit, scuba suit, that does something like that, it may allow for oxygen capture. I would say, yeah, there's quite a bit of engineering ways we could incorporate this stuff too. Anything, Dad?
So was the female larger or smaller?
Females are larger
Females are larger.
in both species, yes.
But they,
Or
but in the Pardosa, it was the Pardosa where they actually time to write was better.
yeah. And then, so in most species, especially the one we compared, the comparable study species, there was a sexual dimorphism in size there. As far as here, I think it's a little bit less, but in Rabidosa, I believe there's more, right? So there's definitely more sexual dimorphism in size in Rabidosa compared to the Pardosa, but there is a little difference there.
Did you do anything where you looked at the relative contributions of each of your factors to time to write?
That's going to be our next study. So we're working on a whole collaborative study where we're going to put everything into a single data analysis and then formulate a figure to do that. So that's next, I guess.
What made you choose these two particular spiders? Are these local to the valley? Your images of the environment didn't seem like it was. And yeah, how did you go about choosing these two?
Okay.
To compare.
Sure. So these species are actually in Arizona in the Chiricahua Mountains. Dr. Stephenson here has actually been kind of doing research there for a while. So he just observed this behavior there, and then those are the two local spiders in that area. So they're close to that research station, and so that's where the spiders were collected.
So females have a lower metabolic rate throughout their lifespan when they're reproducing themselves?
Great question. So actually when they're reproducing, they have a higher metabolic rate just due to reproduction. Whether it's higher than males during that time period or not, I would kind of say so. I don't really know. A lot of the research that I did showed that the time at which they took the submersion tolerance data or any like metabolically related data really was dependent on the breeding season. Because if you have females that are in breeding season, they're going to have a higher metabolic rate, which will influence your results. So with ours, they were in breeding season, right? Yeah. So we
They were
typically see.
in breeding season, they still had a lower
And they still have a low, yeah, so suggests that, yeah, not quite.
Throw that in your model.
Any questions? Thank you guys for your time.
Thanks.